Okay, hello. I want to welcome you to today's RNA Collaborative Seminar Series. For those of you who don't know, I'm Lynne Maquat, and I direct the Center for RNA Biology at the University of Rochester. I also want to thank Liz Leverens for helping to administrate today's event. We have two fabulous speakers today from the University of Rochester. And I want to remind you to press the QA button if you have any questions that arise either during their talks or after their talks. Our first speaker is Dmitri Ermolenko. And for a little bit of background, Dmitri obtained his PhD degree in biochemistry jointly from the Institute of Biochemistry at the Russian Academy of Sciences in Moscow, and also the Department of Biochemistry and Molecular Biology at Penn State University in Hershey, Pennsylvania. He did postdoctoral work with Harry Noller at UC Santa Cruz, and then was recruited to Rochester in 2010. Dmitri is very well known for his single molecule FRET analyses of the structural dynamics and functions of RNAs and of ribosomes during cellular processes, most notably during the process of mRNA translation. And his talk today is entitled, "To What Extent Do mRNA Stem Loops Pause the Translating Ribosome?"
Thank you, Lynne, for a very kind introduction. I'm going to start sharing my slides. So I would like to talk today about the role of mRNA stem loops in translation elongation. My lab studies translation and the ribosome. And we focus on two questions: the role of structural dynamics of the ribosome and ribosome ligands and translation, and the role of mRNA structure in regulation of translation. And the second question stems from the fact that most mRNA sequences have a propensity to form extensive secondary structure. So here in this slide, you see the predicted structure of rabbit beta globin mRNA with the color code that shows a probability of base pairs being formed in this structure. And you can see that there are a lot of stems and stem loops in this mRNA. So how does that affect translation? mRNA during translation is threaded through mRNA channel in the small ribosomal subunit, and this channel accommodates a single-stranded conformation of mRNA. From the first nucleotide of the P side codon to the entry to mRNA channel covers 11 nucleotides. So 11 nucleotides downstream of P side codon, mRNA has to enter an mRNA channel in single-stranded conformation. And as a consequence of that, translating ribosome is an efficient helicase. It was shown by elegant experiments from Harry Noller, Carlos Bustamante, and Nacho Tinoco labs that the ribosome unwinds 3 base pairs at a time per each elongation cycle, per each translocation event. And it's very processive helicase. So if ribosome is a processive helicase, then we would think that secondary structure should not affect much the elongating ribosome. However, despite helicase activity of the ribosome, we know that stem loops can pause the ribosome. For example, during translation of signal recognition particle receptor alpha subunit, a stem loop programs ribosome pause during tethering to the membrane. mRNA stem loops can induce ribosome stalling, no-go decay. And arguably the most well-established example of regulation of translation by stem loops is programmed ribosome frame shifting. And that's what I'm going to focus on. So frame shifting, I'm going to focus on -1 frame shifting, is programmed by 2 sequences. One sequence is a slippery sequence that allows ribosome to slide along that sequence and tRNAs to slide along that sequence without either without introducing mismatches or with minimal number of mismatches, tRNA, mRNA mismatches. And then there is a second sequence that programs frame shifting event. It's called a frame shift stimulating sequence. It's a downstream sequence, either stem loop or a pseudoknot. And it's thought to pause a ribosome, and by pausing ribosome at the slippery sequence, stimulate frame shifting. And I'm going to use two stem loops in today's talk. One is a stem loop from HIV GAG-POL transcript, and the other from DNA-X E. coli transcript. And I'm going to study them. We study them in the context of bacterial ribosome. And we can do that because there were several studies that showed that HIV stem loop produces frame shifting in both bacterial and eukaryotic ribosome. So how do these stem loops perturb elongation cycle? To address that, I would like to remind you the progression of elongation cycle, main steps of elongation cycle. So each elongation cycle begins with peptidyl tRNA bound in the P site, and I'm showing you this a box diagram where the binding sites on each subunit, small 30S and large 50S, are depicted. Then a tRNA arrives to the A side of the ribosome, and ribosome catalyzes peptide bond formation, as a result of which tRNA, P side tRNA becomes deacylated, and peptide is now attached to A side tRNA. Once it happens, the acceptor stems of tRNAs translocate spontaneously from A and P sites to P and E sites on large subunit, while their anticodon stem loops remain in the original A and P sites. And so this hybrid state, AP, PE states of tRNA bindings are formed. And that's coupled to rearrangement of the ribosome. The ribosome with peptidyl tRNA in the P site is non-rotated, but the ribosome with hybrid tRNA in hybrid states is in a rotated conformation, where small ribosomal subunit, shown here in green, is rotated by about 10 degrees relative to the large subunit, shown here in blue. And then EFG, universally conserved factor, protein factor, EFG, or elongation factor 2 in eukaryotes, arrives, binds to the ribosome, catalyzes translocation in a small subunit. tRNAs translocate from A and P sites to P and E sites together with messenger RNA. And importantly, ribosome resets back into non-rotated conformation. So the elongation cycle is accompanied by switching from non-rotated to rotated back to non-rotated conformation. And this has been used in single molecule FRET studies together with FRET studies of tRNA fluctuations to address the question, which step of elongation cycle is perturbed by stem loops that induce frame shifting when they position at entry of mRNA channel. Many studies showed that stem loops, when it's positioned at the entry to mRNA channel, inhibit translocation step. And we also observed that mRNA stem loop position at the mRNA entry channel can also inhibit A side tRNA binding. So structural studies provide some clues of why that could happen. We collaborated with Nico Grigoriev, Sarah Lorsch, and Andrei Koroslev at UMass Medical, and obtained a structure with HIV stem loop bound to the ribosome. To our surprise, a large fraction of ribosomes HIV stem loop binds directly to the A side of the ribosome, forcing partial dissociation of mRNA from mRNA channel, and occupies the A side, partially occluding it for tRNA. So it's probably a transient occlusion that explains the inhibition of A side binding. And then there are several other structures obtained with other regulatory stem loops where the same phenomena is observed, a stem loop bound to the A side of the ribosome. So we think that this could be one of the mechanisms by which stem loops regulate translation elongation. In other structures with frame shifting stem loops and pseudoknots, stem loop or pseudoknot is observed at the entry to mRNA channel with both A side and P side occupied. This is a pre-translocation ribosome, and it's likely that in this structural state, stem loops inhibit translocation. So there are two alternative modes of action. So we wanted to ask, what makes this frame shifting inducing stem loops affecting ribosome posing structures? It's often assumed in the field that the answer to this question is that the stem loops are stable. The ribosome has trouble going through the stem loops. However, the ribosome unwinds only 3 base pairs at a time. So it's also possible that stability of bottom pairs that ribosome first encounters, they are most important. So we wanted to ask a question: to what degree high local and overall thermodynamic stability account for translational pausing mediated by frameshift-inducing mRNA stem loops? And to address this question, we combined this frame-shifting inducing stem loops from HIV and DNAX with non-slippery sequence because we wanted to delineate frame shifting from ribosome pausing. We only want to look at ribosome pausing. And we work with 2 stem loops, HIV and DNAX, and we made mutations in those stem loops. First, we made mutations to destabilize bottom pairs, either 3 pairs in HIV stem loop or 5 base pairs. In DNAX, we destabilize bottom 4 base pairs that are first encountered by the ribosome. We also created a truncated version of HIV stem loop by deleting the upper stem of the stem loop. So the bottom pairs that are first encountered by the ribosome are the same as in wild type HIV stem loop. We also created stem extended stem loop, which is much more stable than the original one. And by the way, the stability, predicted stability of each construct is shown here as delta G, and the changes in stability relative to the wild type are shown as delta delta G. And finally, we changed the loop sequence, extended loop sequence in both HIV and DNAX constructs to see contributions of the loop sequence to the pausing. And in addition, we made bimolecular helices. Essentially, we truncated stem loops, cleaved it in the middle, and annealed, recreated the stem by annealing RNA oligo. So we have this loopless bimolecular helices. For the sake of time, I'm not going to go through experiments where we test the effect of these constructs on tRNA binding. Like I told you, these stem loops can do both things. They inhibit tRNA binding and inhibit translocation. The summary of these experiments are shown here. The changes in stability didn't affect much the ability of stem loops to inhibit A-site tRNA binding. Truncated stem loop showed weaker inhibition of A-site binding. Extended stem loop and bimolecular helices were not able to inhibit A-site binding at all. And it sort of makes sense because it's that the length and the structure of a stem loop plays the key role in inhibiting A-site binding, because that probably is important for docking into the A-site. The shorter stem loop has a lower ability to inhibit tRNA binding and compete with tRNA for binding to the A-site. And the extended stem loop probably doesn't fit into the A-site, and bimolecular helices don't bind into the A-site. And for that reason, they don't inhibit tRNA binding. But I'm going to focus more on the translocation, inhibition of translocation step, because it's likely that this is more common and more universal mechanism by which secondary structure perturb translation. So we wanted to see how these variants of stem loops perturb the rate of translocation by following switching from rotated pre-translocation conformation of the ribosome to non-rotated conformation, which occurs during EFG catalyzed translocation. To follow intersubunit rotation, we used Forster resonance energy transfer using FRET assay that was established in Knowler lab, where we introduced fluorophore and the protein L9 of large subunit, and fluorophore, the other fluorophore, acceptor fluorophore, and protein S6 of small ribosomal subunit. And as you know, in FRET, when dyes are closer in distance to each other, then their energy is transferred from blue shifted donor to red shifted acceptor, and there is some more acceptor fluorescence, less donor fluorescence. When distance between dyes increases, efficiency of energy transfer drops, so we see more donor fluorescence and less acceptor fluorescence. And so we can measure single FRET in individual ribosomes doing single molecule FRET measurements using total internal reflection microscopy. And a typical trace from one ribosome is shown here, where the donor fluorescence is shown in green, intensity of acceptor fluorescence is shown in red, they anti-correlated, and then resulting FRET is shown in blue, and you can see that FRET fluctuates between lower 0.4 FRET state, which corresponds to a rotated conformation of the ribosome, and higher 0.6 FRET state, which corresponds to non-rotated conformation of the ribosome. So now we're going to position stem loop at the entry to mRNA channel in the pre-translocation ribosome with peptidyl tRNA bound on the A side and deacylated tRNA on the P side. And we're going to start imaging this ribosome. So here's the typical trace from one that we obtained in one ribosome. We start imaging, and at 10 seconds after the beginning of imaging, we're going to inject EFG and GTP and measure time it takes for the ribosome to switch from 0.4 rotated state to 0.6 non-rotated state. And this is time that corresponds to rate of translocation. So when we position HIV wild-type stem loop at the entry to mRNA channel and inject EFG at 10 seconds, here's the typical time trace that we get. Blue here is intensity of donor, red is intensity of acceptor, and resulting FRET is shown in yellow. So it takes quite a bit, over a minute, quite a bit of time for the ribosome to switch from 0.4 to 0.6 FRET state. We can then aggregate the data from multiple time traces and plot distribution of translocation times shown here on the right. And you can see that the median time, translocation time is over a minute, is 73 seconds. When we do control experiment with mRNA that doesn't have stem loop, we observe much faster translocation. If we look at distribution of translocation times, the median time for translocation is 3 seconds. So the stem loop position 11 nucleotides downstream of P side codon right at the entry to mRNA channel, inhibits translocation, produces 20-fold reduction in translocation rate. And this effect depends on the spacing between the stem loop and the P side codon. The further the stem loop from the entry to mRNA channel, the less inhibitory it is. So right at the mRNA channel, translocation is 73 seconds, medium translocation time. When we move stem loop 12 nucleotides away from the P side codon, translocation is now, it takes 30 seconds, medium time. Here's the distribution for this construct, for this conditions. And finally, when we move it 13 nucleotides away from a P side codon, now translocation median time is 17 seconds. So much less than the 73 seconds that we observed at 11 nucleotide spacer. So now we're gonna look at the different mRNA variants positioned 11 nucleotides downstream of P side codon, where we observe the maximum of inhibition of translocation. I'm going to start with, this graph shows the median translocation time for different constructs, and I'm going to start with the control that doesn't have stem loop for both HIV variant and DNAX variant. In the absence of stem loop, translocation is fast, 3-5 seconds median time. Then with the wild type stem loops, both HIV and DNAX, we observe close to 20-fold inhibition of translocation. Now translocation time is over a minute, 73 seconds. When we change the loop sequence, really nothing happened. Translocation remains slow, 70 seconds for both HIV and DNAX. When we destabilize bottom pairs, either 3 pairs for HIV or 5 pairs in HIV, we saw substantial reduction in translocation time. Now it's faster, 41 seconds, 32 seconds for 5 nucleotide variant, where 5 base pairs are destabilized. Definitely, destabilization of base pairs that are unbound by the ribosome makes a difference, but still translocation is 10 times slower than translocation in case of no stem loop control. And we observe similar things for DNAX construct. To our surprise, when we made truncated stem loop and extended stem loop, we observed only 3-fold reduction in translocation rate relative to no stem loop control, 10-second median translocation time. And it's puzzling because the stability of HIV stem loop, extended stem loop, is much higher overall stability than the wild type sequence. And the identity and stability of bottom pairs and HIV truncated, HIV extended constructs is the same as in the wild type. We obtain similar results with biomolecular helices. Bimolecular HIV helix in DNAX bimolecular helix inhibit translocation only 2 to 3-fold. Now we have translocation time of about 10 seconds. So again, interestingly, that the identity of base pairs first unwound by the ribosomes are identical in bimolecular helix, extended stem loop, truncated stem loop, and wild type HIV stem loop. So we think that this shows us how much unwinding of 3 base pairs slows down ribosome. That these results are also consistent with optical tweezers experiment that suggests the same thing, that translocation along 3 consecutive base pairs slows down ribosome only 2-3 times. By contrast, the wild type stem loop inhibited translocation much more, because the length and the structure of that loop are more important than thermodynamic stability of bottom pairs. And we think this is because the stem loop interacts with the ribosome. So I think our results taken together suggest that the stability is not the main factor. Stem loops don't act as simple barriers that ribosome has struggled to unwind. They interact with the ribosome in a way that inhibit translocation much more than just unwinding of 3 base pairs. One proposal put forward by Needed Bun, based on his reconstructions of the pseudoknot from SARS-2 bound to the ribosome, is that pseudoknot, in his case pseudoknot, but it could be the same for stem loops, interacts with the small ribosomal subunit in a way that it interferes with rearrangement of the small subunit, the rotation of head domain of a small subunit that is coupled to translocation. And by doing this, stem loops or pseudoknots of specific length and structure enable long pauses. And finally, I told you that we did all these experiments in the context of non-slippery codons. How does frame shifting efficiency correlate with all those pausing rates that we measured in non-slippery mRNAs? So we recreated some of those constructs in the context of slippery sequence in DNAX mRNA and measured frame shifting efficiency by filter binding, tRNA filter binding assay. So the slippery sequence alone, in the absence of the stem loop, produces 18% frame shifting, quite high level. And then wild type intact DNAX stem loop produces additional 3-fold increase in frame shifting efficiency to 61%. And that's consistent with the literature data that were published previously. When we destabilize bottom 4 pairs in DNAX stem loop, the frame shifting efficiency drops from 61 to 41%, but we observe almost complete elimination of the effect of the secondary structure when we replace the stem loop with the bimolecular helix, which produced much less significant ribosome pausing in our experiments. So you can see that the pausing times that we measured in single molecule experiments shown here roughly correlate, at least to a degree, with the frame shifting efficiency that we measured in this limited data set. So circling back to the first question that I asked, to what extent mRNA stem loops pause the translating ribosome? Our results suggest that translocating to 3 consecutive base pairs is only 3 times slower than translocation along single-stranded mRNA. This is the slowdown of translocation that we observed with truncated and extended stem loop and bimolecular helix. And we've done some experiments where we just looked at duplexes formed by oligonucleotides and mRNAs and observed a similar phenomena, 2 to 3-fold decrease in translation. Of course, if you have multiple consecutive slowdowns of this magnitude, we can have cumulative effect as ribosome translates through extended secondary structure, but it is not going to pause for a long time. And then frame shift inducing stem loops of specific length and structure induce much more significant 20-fold reduction in translocation rate, likely because they make specific interactions with the ribosome. And finally, I would like to thank people who did all the work. This work was spearheaded by a talented PhD student in the lab, Chen Bao, who did all the biochemical and single molecule experiments with contributions from Hiro Wakabayashi, Clarence Lynne, and Ina Nikonchuk in the lab. Dave Matthews and Mingyi Zhu helped us designing the mutants and predicted the thermodynamic stability of mutant stem loops. And finally, Sarah Lorsch, Nico Grigoriev, and Andrei Korostelyov determined the structure of a ribosome with HIV stem loop. So I also would like to thank you for your attention, and I'll be happy to answer your questions.
Thank you, Dmitri. That was pretty interesting. So you have a couple of questions. Danielle, would you please type your question into the Q&A box? And in the meantime, Ilya Taranin has a question. Thanks for the talk. As for a sign just went up over the question. Oh, here. As for the stem loops that are observed in the A site, how can they even enter a ribosome? Shouldn't these sequences be unwinded in order to enter the channel?
So, of course, we don't know precisely how it enters the A site, but the segment of the sequence between the P site codon and where it should have come out of mRNA channel is dissociated from mRNA channel. So this has to, the docking of the stem loop has to be coupled with partial dissociation of mRNA from mRNA channel. But the stem loop is intact.
Okay. I see Danielle's question now. How does the stalling of the ribosome on the stem loop affect the mRNA stability and decay rate? Does the stem loop ever get linearized? And if so, does it reform and stall the next ribosome in line? Wong et al. Showed in 2022 that initiating ribosomes oscillate around stem loops. Does this happen here?
Well, this is elongation phase, so it's going to be a little bit different from initiation, but we didn't measure mRNA stability. It could be that the pauses that are observed in vivo conditions where the concentration of elongation factors are much higher, the pauses are a little bit less dramatic, and it's more of a transient event. So it might not trigger mRNA decay, a no-go decay, or something along those lines. And yes, as ribosome plows through it, unwinds stem loop, and the stem loop might be reformed, and the lagging ribosome might encounter this again. But it's a dynamic thing that the stem loops unwound and reformed over and over again.
There is a question from Daniel Alberts. Isn't the speed 15 amino acids per second? Yours is 1 amino acid per 3 seconds. Is it impeded?
Right. Our rates, like I said, the in vivo rates will be much higher because the concentration of elongation factors is insanely high in vivo. It's a 100 micromolar range for EFG, both EFG and EFTU. So the rates in vivo will be higher, and there were some in vitro systems reconstituted that show higher rates by optimizing conditions. But so the sum of the effects we observe might be a little bit less pronounced in vivo.
Okay. And then I have one last question. So Dmitri, when you delete the FSS and you report 18% frame shifting just with the slippery codons, were they in the -1 frame? Was all frame shifting in -1 frame?
We only measured -1 frame shifting. I cannot say whether any other type of frame shifting.
Okay. I think we need to move on. So there's an anonymous person asking a question, and I'm sure Dmitri will be happy to answer that for you in the near future. Thank you, Dmitri. That was fabulous.
Thank you.